Hello, this is Unit 2, Cell Structure and Function. So we're going to begin with talking about the similarities between the eukaryotic cell and the prokaryotic cell. To begin with, both cells have DNA. This is the genetic material that they use. In addition, they both have a plasma membrane. The plasma membrane surrounds both cells. They have cytoplasm, that is the stuff inside the cell, and we call the liquid portion of this, or the fluid portion of this, the cytosol. And last but not least, both the eukaryotic cell and the prokaryotic cell have ribosomes. Both of these cells need to synthesize protein, and they use a similar mechanism for both. So what is the differences between these two cells? Well, to begin with, the prokaryotic cell has circular DNA, where the eukaryotic cell has linear DNA, so the chromosomes are linear structures. Additionally, the eukaryotic cells have a nucleus, and they have membrane-bound organelles, such as the mitochondria, the chloroplast, Golgi, or ER. Next, we are going to look at the individual organelles that are present in the eukaryotic cell. We'll start with the nucleus. The nucleus is center control of the cell. This is where your DNA is found. The purpose of the nucleus is to basically protect your DNA from mutation and other damages that could occur. Interestingly, surrounding the nucleus, which is the nuclear envelope, there are the presence of these little holes that we call pores, and the pores control what enters and leaves the nucleus. Our next stop are the ribosomes. The ribosomes are the protein synthesis factories of the cell. This is where they get the mRNA, and those are the instructions for synthesizing protein. There are two forms of ribosomes, the free ribosomes, and those ribosomes are going to be floating around in the cytosol of the cell. The second type of ribosomes are the bound ribosomes, and these ones are exclusively attached to the ER. In particular, these are proteins that are important or essential for export. So proteins that we secrete from our cells are going to be synthesized by these ribosomes and not the free ribosomes. The free ribosomes are the proteins that are used within the cell. Our next stop is the endoplasmic reticulum, or the ER. Notice how the ER is located just outside of the nucleus. In fact, this membrane structure really seems as if it's part of the nuclear envelope. There are two types of ER. The first type is the rough ER, and the rough ER is rough because it has the ribosomes present on that, hence the name rough ER, or ribosomal endoplasmic reticulum. The function of this ER is to package proteins that are important for secretion. After they are packaged in the ER, they are then shipped to the Golgi, where more packaging and more folding and other modifications take place. The other type of ER is the smooth ER. So this ER does not have any ribosomes associated, so therefore it has absolutely no role in the synthesis of proteins. Instead, what the smooth ER does is it helps synthesize lipids, and it acts to detox drugs and other poisons that may enter the cell. The key thing for you is this idea of synthesizing the lipids. So make sure we know that the rough ER is important for protein production, where the smooth ER is important for synthesis of lipids, but has nothing to do with proteins. Our next stop is the Golgi apparatus. Think of the Golgi apparatus like the UPS shipping department. The purpose of this organelle is to process and package material. So it arrives in vesicles. If it's a protein, there may be folding involved, modification, and then it is packaged in these vesicles and then shipped elsewhere in the cell. So you can see these excretory vesicles here that are fusing off of the Golgi apparatus. Vacuoles, and I should also include vesicles on here. The job of vesicles and vacuoles is to either store material, such as food and water, or they are also going to be necessary for transport of material. So they are going to be important for moving material from the ER to the Golgi and from the Golgi to the cell membrane. The next organelle are called the lysosomes. The lysosomes are unique because they have these digestion enzymes within them. And so these enzymes are released into vacuoles, or food vacuoles, and what they do is they help in the digestion of macromolecules that enter the cell. One of the important things that you'll need to know for your test is called the endomembrane system. The endomembrane system is important for regulating protein traffic and performs other metabolic functions. Basically, I like to call this the protein assembly. So we start here in the nucleus. The nucleus is where the DNA instructions are kept. We make mRNA. The mRNA is then exported through the nuclear pores and then arrives at these ribosomes. The ribosomes are where we synthesize the new protein. The new protein is then enters the ribosome where we have some processing that take place. It then travels from the ER to the Golgi. From this organelle to this organelle, make sure that you remember that there are vesicles involved in that process. In the Golgi, we have more folding, we have more modification. It is then again packaged in a vesicle and will then be exported from the plasma membrane into the extracellular surface of the cell. Here's just another way of looking at it. So you can see that our proteins would start here in the nucleus. Our protein production starts in the nucleus. The mRNA travels out through nuclear pores. The mRNA then is used as instructions to synthesize protein in the ribosome. These are the rough ER because they have the ribosomes attached to the ER. That protein then is fed through the ER, is then packaged in a vesicle. The vesicle then moves from the ER to the Golgi, where more modifications and folding happens. Within the Golgi packs it in a vesicle, and that vesicle then is transported to the cell membrane, where it is secreted by the process of exocytosis. So this is an important pathway for proteins that are secreted from the cell. Two other organelles, and these are ones that we'll predominantly be talking about in the next unit, are the mitochondria, which is necessary for cell respiration. So the other thing with regards to the mitochondria is this helps the production of ATP. So ATP is the energy source of the cell, and so if you see anything about ATP production, that is going to be the mitochondria. The chloroplast is where photosynthesis happens, and so if you see anything about the production of glucose, that is going to be the chloroplast. Chloroplasts are exclusively in plant cells and not animal cells. This right here just is the review of the different organelles that we talked about. So you can use this for review purposes later. All right, now we want to talk about the idea of cell surface area. As we discussed in class, cells must be small to maintain a large surface area to volume ratio. That is the SA to V ratio. On the test, you may be asked to calculate this. I will give you the formula. You can use your calculator. Remember, when you are comparing two cells, always go with the cell that has the largest surface area to volume ratio. That is because that cell is able to move molecules in and out of its membrane most efficiently. And that is really demonstrated here below. You can see our small cell right here. That is a 1 by 1 by 1 cube. If we calculate the surface area of that, it's 6. If we calculate the volume of that molecule, it's 1. 6 divided by 1 is a value of 6. Where if we look at a larger cube here, a 5 by 5 by 5, even though it's larger, it has a larger surface area, it has a larger volume, but when you divide these two, you end up with a surface area to volume ratio of 1.2. Notice that the 1.2 is much smaller than the 6. Even though this cell right here is smaller, it is more efficient at moving the necessary material in and out of the cell. So that could include raw materials such as food, water, or oxygen, and the removal of waste or carbon dioxide. And so you can see right here that rather than have a large cell such as a 5 by 5 by 5, we can decrease the surface area, so basically take this 5 by 5 by 5 and break it up to 125 individual cubes, our surface area dramatically increases. However, our total volume doesn't. And so because we increased our surface area to area to volume ratio is back at that 6, which is much higher than the individual cube and is much more in line with the smaller cube. So breaking our cube up into mini makes for a more efficient exchange of chemicals between the environment and the cell. So on your test, always look for the example with the highest surface area to volume ratio. Because if that surface area to volume ratio becomes too small, it's going to decrease the rate of chemical exchange, so the inside and the outside, and that could cause the cell to die. The next topic we looked at was the cell membrane. Remember that your cell membrane or plasma membrane is selectively permeable. That means that it allows certain substances to cross, while others it does not. We also describe our cell membrane as a fluid mosaic model. So let's break down the two words as this descriptor. Our first one is fluid. That's because the membrane is held together by weak interactions. So those individual phospholipids, they interact with one another, but their interactions are relatively weak. And so think of the individual phospholipids as having the ability to freely move about the cell membrane. The next descriptor in our model is called mosaic. And that's because the cell membrane is made up of phospholipids, proteins, as well as carbs, and some cholesterol. And so the cell membrane is this mosaic of many different things that make it up. The major component of the cell membrane is called the phospholipid. Phospholipids are unique molecules in that they have a hydrophilic head, and this head is attracted to water. And they have a hydrophobic tail, which is repelled by water. So we actually call these molecules amphiphilic. When you have a bunch of these molecules stuck together, they form the bilayer. You notice in our bilayer here that our hydrophilic heads are pointing outward towards the water. So this would be the extracellular side, and our phospholipid heads are pointing outward towards the water and our intracellular side. Water on the outside, water on the inside. Whereas our hydrophobic tails right here in the middle, they are pointing towards one another. This is because we have the exclusion of water in the middle of our cell membrane. In addition to phospholipids, we also include cholesterol. The job of cholesterol is multifold. Cholesterol basically resists changes. So at high temperatures, it limits fluidity, and at low temperatures, it inhibits the ability of the phospholipids packaging too tight. So cholesterol functions to reduce membrane fluidity and moderate the temperature. In addition to cholesterol, there are many proteins embedded within our cell membrane. The ones that go through the membrane from one side to the other are called integral proteins. These are embedded within the membrane. Whereas the peripheral proteins, these are sitting on the surface of the cell membrane. So they may be on the extracellular side, or they may be on the intracellular side. The transmembrane proteins, or the integral proteins, have very unique properties. You notice that the ends here, these are the hydrophilic ends, they are going to be pointing out to the water, because there's water here on the intracellular side, and there's water here on the extracellular surface of the cell. In addition, they're going to be interacting with these hydrophilic heads of our phospholipids on both the extracellular surface as well as the intracellular surface. However, in the inside here of our transmembrane protein, this is where our hydrophobic amino acids are going to have an intricate relationship with the hydrophobic tails of our fatty acid chains on our phospholipid molecules. As I mentioned, the cell membrane is selectively permeable. So what does that mean? Well, it means that small nonpolar molecules such as oxygen, CO2, or N2 are freely able to move from one side of the membrane to the other. They are able to move past those phospholipids. Whereas larger molecules, polar molecules, or ions aren't able to move. They try to go through that phospholipid bilayer, and then right here in the middle, this hydrophobic barrier blocks their access and causes them to basically leave the area. The only way in which these molecules, polar molecules, such as glucose, or ions such as protons, sodium, potassium, can enter is that they're going to have to move through one of those embedded channels. And I'll show you that in the next slide. Now, I do want to make the statement regarding water. Water is a polar uncharged molecule. It is able to pass in small amounts. So some water is able to leak through the membrane. Not a lot, but there is definitely a little that is able to leak through. As I said in the last slide, these hydrophobic substances, such as large polar molecules and ions, they must move through a membrane channel. So that embedded protein acts as a channel that allows specific molecules that wouldn't normally be able to cross the membrane to now be able to enter the cell or leave the cell. This includes polar uncharged molecules such as water that's able to pass with a little amount. When we talk about movement in and out of the cell, we need to talk about diffusion. Diffusion is the movement of molecules from a high concentration to area of low concentration. So you can see in this example here, we have a highly concentrated amount of molecules, and then over time it's going to spread out and fill that space within the beaker. Moving things into the cell, we first look at passive transport. Passive transport is moving with the gradient, diffusion down the concentration gradient. So in this situation, we're always going from a high concentration to a low concentration. It requires no energy. So no ATP is necessary for this. And we call this passive transportation. A form of passive transportation includes facilitated diffusion. Facilitated diffusion relies on the assistance of a transport protein. Basically, this is one of these proteins that are embedded within the cell membrane. And they provide a bypass for molecules that would normally be able to pass the cell membrane to go from one side to the other. So they bypass the hydrophobic core of the cell membrane. When we discuss the term facilitated diffusion, facilitated just means that it needs a channel or a carrier protein. And diffusion is still traveling from a high concentration to a low concentration. Once again, diffusion down the concentration gradient. No energy is necessary during this procedure. When we have facilitated diffusion, this also allows us to create what we call a polarized membrane. So you notice in this example right here, sodium has a positive charge. And so we can compare the number of sodium ions outside the cell versus the number of sodium proteins inside the cell. When we start off with, we have a whole bunch of sodium proteins outside the cell. So we assume that outside the cell is going to be positive 8, where inside the cell we only have one sodium ion. And so we say that it is positive 1. Now if you subtract these two, or make a relative term, so that the outside of the cell versus the inside of the cell, we can assume that in this example right here, we have a -7 inside the cell. One of the important pumps to establish a membrane potential is the sodium potassium. The way this pump works is it pumps sodium from inside the cell to outside the cell, building a concentration gradient. Similarly, the same pump pumps potassium from outside the cell inside the cell. Both the inside and outside are going in the opposite direction, so they're going against the gradient. So this is going to require energy. This is an example of active transportation. So what is active transportation? Well, active transportation requires energy. That's because we are going against the concentration gradient. So we are going from a low concentration to a high concentration. In a question, you'll see also the word pump. So if you see pump, if you see the necessity of energy, such as ATP, or going against the gradient from low to high, in all these situations, we're discussing the same thing. This is what we call active transportation. In this slide, you can see the compare and contrast between passive transportation and active transportation on the right-hand side. Passive transportation requires little or no energy because we're always going with the gradient or down the concentration gradient from high to low. Examples of this include diffusion, osmosis, facilitated diffusion, whereas active transport requires energy. That's because we're going against the concentration gradient. We are moving from a low concentration to a high concentration. So things such as pumps and then this process called exocytosis or endocytosis are also examples of active transport. So to summarize the different forms of transport, we can look at this diagram here. We start off with simple diffusion. So simple diffusion is simply moving across the membrane without any assistance. We are always following the concentration gradient because we're going from a high concentration to a low concentration. So no energy is necessary for this to take place. The next type of movement is called facilitated diffusion. Facilitated means that we need a specific carrier protein to accomplish this task. However, because once again, we're going from high concentration to low concentration, we can assume that this is also a form of passive transportation. That's in contrast to on the right-hand side of the screen here, where we have active transportation. Active transportation is going against the concentration gradient. So in this situation, we're going from a low concentration to a high concentration, the opposite that it should. And because we're going against the concentration gradient, it is essential that we are using ATP to drive this pump. Now, that's all fine and dandy for molecules that are smaller, but when we have larger molecules, this is what we use for what we call bulk transport. So this includes proteins, polysaccharides, and other large molecules. There are two processes. The first process is called endocytosis. Endo means to enter the cell. Notice how that our cell membrane forms a vesicle around the material that it wants to enter the cell. That vesicle pinches off and then it moves into the cell. Exocytosis is the same process in the opposite direction. So in this situation, we have our vacuole here, or our vesicle here. It moves towards the cell membrane and fuses with the membrane. Eventually, it dumps out as cargo into the extracellular space. Remember, endo is to enter, exo is to exit. Next, we are going to look at osmosis, which is the diffusion of water. Remember, water always goes from the low concentration of solute to the high concentration of solute. Or remember that salt sucks, or salt pulls the water to that side. These are examples of U-tube models. I remember in these U-tube models that we have this membrane right here. It's a selectively permeable membrane. And so there's these little gaps in that membrane. And what that does is it allows water to either move to the left or to the right. However, if you're bigger molecules like this green thing right here, we can assume that green thing is sugar. That sugar is too large to move back and forth through that membrane. In this slide right here, you will see the different ways that we can describe the movement of water across a selectively. Remember first and foremost that water always diffuses from a high concentration to a low concentration. A dilute solution to a concentrated solution, remember because the solute is going to pull it that direction. It always goes from a high water potential to a low water potential, or from a low osmolarity to a high osmolarity. So there's lots of ways of describing the movement of water, but it always goes from a high water potential to a low water potential. Next, we have here the three scenarios: hypotonic, isotonic, and hypertonic. When we look at the hypotonic solution here on the left-hand side, hypo, like a hippo, that's because on the outside of the cell, there is a lower concentration of solute. Therefore, there's a higher concentration inside, and it's going to suck the water into the cell, causing the cell to swell and possibly erupt. Whereas the plant cell it has this big central vacuole and it fills with water. However, outside of the plasma membrane is the cell wall, and that protects the cell from really exploding or lysing, such as an animal cell. And so in this situation, we would assume that this cell is what we call under turgid pressure. Now on the right hand side of the screen here, we have the hypertonic, hyper like a viper, because the cell is going to shrink and water is going to leave the cell. Hypertonic because we have a higher concentration of solute outside the cell. Therefore, water goes from the high potential on the inside to the low potential on the outside. And in the middle here, we have the isotonic solution. That means that inside the cell is the same as outside the cell. We still have water movement, but there is no net gain of either or loss of water in the cell. Certain organisms need to deal with osmoregulation. One of the examples that we see in biology is the paramecium. So paramecium live in fresh water, and so you can assume that outside the cell is a hypotonic solution, and inside the cell we have lots of solute. So because this is hypo on the outside, water is going to move into the cell and would cause the cell to explode. However, instead it has this thing called a contractile vacuole. And so that vacuole gets bigger and bigger and bigger, and then squeezes and then pushes the water back out of the cell to prevent it from rupturing. When we talk about water, we have to deal with water potential. So this is the equation for water potential. It has the solute potential represented by psi s and the pressure potential represented by psi p. When we add these together, that is what we call the water potential. Remember that water always goes from a high potential to a low potential. Fresh water or pure water has the highest concentration or the highest water potential that we call 0. Adding solute to that water decreases this potential, and therefore it becomes more negative. So when we talk about water potentials, a lot of the times what we're dealing with is we're looking at negative numbers. So don't get mixed up with that. As we add more solute, the solute potential becomes more negative if it's not pure water. When we are comparing the movement of water, water always falls from high water potential to low water potential. So it always goes towards the more negative. Be careful about that. Students always get those negative numbers mixed up. It always goes towards the more negative of the two water potentials. Because remember, 0 is our highest potential. So anything else is going to be negative, and more negative. Some examples of how this works is we have 3 U-tubes. On the left-hand side, or example A here, we have a fresh water tube. Therefore, the solute potential is 0. And this is an open tube, so our water potential is, or the pressure potential, is also 0. We add those two together with a result of 0 for water. On the right-hand side, however, here we have solution. And so that gives us a negative number for our solute. Add our solute with the 0 pressure, we have a negative value for our water potential. Remember, water always goes from the higher, which is 0, to the lower, which is -4. So it's going to go from the left to the right. In our middle example here, we've actually added some pressure to our tube. So we're pushing down upon our tube. We have a plunger right here. And so what that plunger does is it adds pressure to our tube. So we represent that by the plus 0.15. Add that to the negative solute, and we end up with a value of 0. The 0 here is equal to the 0 here. Therefore, there is no net movement of water from either the left or the right hand side. Similarly, we add the same situation on this side. This time, we're adding a value of 0.4 and a solute potential of -4.4. We add those together for a value of 0. And once again, just like the middle example, we don't see any movement of water. So that's if I give you the solute potential, but you also have the ability to calculate the solute potential. And that is using this formula right. Psi s is equal to -ICRT I represents the ionization constant, and so that's the number of particles made in water. I will give you this number. Glucose, sucrose, those are all an ionization number of 1, whereas sodium chloride is an ionization number of 2. So just read the question, it will tell you what to do. C is the molecular concentration. Once again, that's going to be given in the question. R is the pressure, that's a constant. I will give you that constant, you just plug it in. And then lastly, T is the temperature. Now just be careful with the temperature because we work in kelvins. So that means you have to add 273 to whatever degrees Celsius temperature I give you. So for example, if I tell you the temperature is 25 degrees Celsius, you add that to 273. And so your T value would be 298. Plug that into your calculator, and that will give you your solute potential of whatever you're looking at. Remember that as you add more solute, the potential becomes lower or more negative because you're decreasing the water potential. One thing I should note is don't forget that negative sign. It's one thing that a lot of students forget to include. So here's an example of how you would calculate the solute potential. Here's our formula, negative ICRT. In this example here, we have 1 mole of sugar at 22 degrees Celsius at standard atmospheric conditions. So sugar has an ionic value of 1. Our molarity is 1. Our R constant is 0.083. And our temperature is 22 degrees Celsius plus 273. Gives us a value of 295 Kelvin. Plug that into the calculator, and you end up with a value of negative 224.22, and our unit is bars. So on your test, make sure you're able to calculate cell compartmentalization. When we deal with cell compartmentalization, there are 3 reasons why we have compartments, i.e. organelles, in our cells. They provide an advantage compared to bacterial cells. The basic understanding is that they increase the efficiency by which the way that our cells function. These roles include establishing physical barriers, generating specific microenvironments, such as the lysosome, and lastly, they provide a distinct cellular address for which cells are able to process different biological reactions, such that organelles have very specific roles. Just as the mitochondria is important for producing energy, whereas the Golgi is important for causing proteins to fold. Next, the importance of the intercellular membranes or internal membranes. And we see these in the mitochondria, we see these in the Golgi, and they have 2 important roles. The first job here is to minimize the competing interactions. So as I said before, the different organelles have different functional roles. So you don't want the mitochondria and the ER competing for their different jobs. And the other reason why having internal membranes is critical is it helps increase the surface area for reactions. And that's really important in the mitochondria and the chloroplast for generating glucose or ATP. And to finish off, the importance of or evidence to support endosymbiosis. So it is believed that a few billion years ago, our prokaryotic ancestral cell decided that it was going to infold its cell membrane. As it infolded, we eventually created the nuclear envelope, as you can see here, as well as the ER and the Golgi. Then a few thousand, maybe million years later, our prokaryotic cell decided it was going to eat another prokaryotic cell. Rather than chew it up and swallow it, it actually formed a symbiotic relationship. And this is the origins of our mitochondria. Now, our plant friends went one step farther and they engulfed a second prokaryotic cell. And that cell now has become the chloroplast we see in plants. Scientists believe that this is the scenario by which we acquired these 2 organelles. And evidence to support this theory are 4. The first piece of evidence to support the endosymbiosis model is the presence of a double membrane on our mitochondria and on the chloroplast. It is believed that this second membrane appeared through the process of cytosis. The next evidence is how these organelles divide. They divide in a process called binary fission. This is similar to the way that bacteria cells also divide. The next piece of evidence, which I consider the most strengthening, is the presence of DNA. Yes, the mitochondria in the chloroplast have its own DNA. Its DNA is a circle similar to that found in bacteria cells and not linear like our cells. And the last and final piece of evidence is the presence of ribosomes. And these ribosomes are smaller like you would see in a bacteria cell and not like our cells. So that is the end of the review. Best of luck on your test. Study hard.